La siguiente disertante es Melissa Landrum. Aquí la tenemos. Bueno, es científica. Melissa forma parte del personal científico de la Biblioteca Nacional de Medicina del NIH y es jefa del equipo Clinvar, que es una de las bases de datos genómicas públicas más importantes a nivel mundial, en donde se publican las relaciones entre las variantes genéticas y el fenotipo humano. Por motivos personales, Melissa no puede estar presente para poder responder preguntas, pero envió su presentación grabada, que se titula Datos e integración de Clinvar en historias clínicas electrónicas. Así que bueno, ahora les vamos a mostrar la presentación que nos envió. A ver si podemos compartir.
Good morning, and thanks to the organizers for giving me this opportunity today to talk with you about Clinvar data and how it can be integrated with EHRs. So let's start with some background about the challenge of variant interpretation. So as many of you know, next generation sequencing now makes it possible to test whole genes, whole exomes, or even whole genomes as part of clinical genetic testing. But the result of this is that now clinical genetic testing laboratories not only identify known pathogenic variants, they're identifying many novel variants. And these novel variants require that the lab do research and collect evidence to evaluate what the clinical significance is of these new variants. So this is complicated because many of these variants are rare. The lab hasn't seen them before. They can query databases of variants to see if others have seen them, but many of these databases are proprietary or require subscriptions. There's an additional challenge that even when different groups use the same set of rules for classifications, they may come up with a different interpretation. So you really need to know why they classified a variant as they did in a specific case. And of course, all of this is very important because the ability of clinical testing labs to provide high quality interpretations of variants on lab reports is very critical for physicians to give good patient care. So with that in mind, we have the Clinvar database, which is NIH's database of interpretations of genetic variants relative to conditions. And it's a way for clinical testing labs and others to share their interpretations and their evidence to improve everyone's ability to interpret variants. The interpretations that you see in Clinvar are considered variant level. So in other words, we're not talking about an interpretation of what a variant means for a patient's own phenotype, but rather a more broad interpretation of what a variant means for a disease. The database is driven by submissions of interpretations from the community, and all of the data is public and available for any use. Today, the database holds more than 1.8 million interpretations for over 1 million variants. And these data come from more than 2,000 submitters from 82 countries across the world. So Clinvar's main goal is to help clinical genetics testing laboratories and the clinicians who receive those lab reports research the variants that have been identified in clinical testing. So there's two major use cases that we think about. One is from the perspective of the clinical testing lab. So let's say your lab has identified a novel variant. As you research it, some of the questions you'll ask are, have other labs seen this variant? How did they interpret it? And what was their evidence for the interpretation? A second use case is more from the clinician's viewpoint. So a clinician may have a patient who has results from genetic testing, and maybe there was a variant reported as pathogenic or a variant of uncertain significance. And that physician may need to ask questions like, how have other laboratories reported this variant? What phenotypes were present in other individuals with that variant? And is there new evidence for this variant since my patient was last tested? And Clinvar can help answer all of these questions. So to give you an overview of the kind of data you see in Clinvar, we're going to take a look at one of the Clinvar variant pages. So right at the top of the page in this dark blue bar, we show you the variant that's represented by this page, followed by some overall information like the overall interpretation of the variant and the review status. We'll talk more about these fields. There's also a variation ID, which is a unique identifier that Clinvar assigns to each variant. And then the tabs on the left provide you with more details about the variant, the conditions reported for the variant, and the affected gene. As you scroll down the page, you get more specific information about each submitted interpretation. So in this table, each row is a submission from an individual laboratory. There's 3 labs who've provided information about this variant. And for each submission, you can see what their interpretation of the variant was, their assertion criteria or the rules that they use to classify variants, the disease that they classified the variant for, and then sometimes a comment or citations that support their interpretation. The scope of data that you'll find in Clinvar is pretty broad. So the variants can be anywhere in the genome. We include both short variants like SNVs and indels, as well as larger variants like copy number variants. Most of the interpretations in Clinvar are for single variants. Occasionally, it's important to interpret 2 or more variants as a haplotype or genotype, but most of the time, records are for single variants. The conditions you'll see represented in Clinvar include diseases, phenotypes, as well as drug responses. Again, typically variants are interpreted for a single condition. Occasionally, there's a variant that's interpreted for a combination, and that is most typically when a variant has been interpreted for a new Mendelian disease that is not yet named, but is described by a set of phenotypes or clinical features. And then for the interpretation, you'll find variants that are interpreted for their pathogenicity for Mendelian diseases, their relationship to cancer, and for pharmacogenomics. So as of November 5th of this year, 2,062 organizations have shared data through Clinvar. Most of the data that you see in Clinvar are from clinical genetics testing labs, but we also have data from research labs and locus-specific databases. We have some data from clinical providers and patient registries. These submissions typically focus on phenotypic data, which is information that the clinical labs typically don't have. We also have variants from OMIM and gene reviews, as well as variants that were reviewed by expert panels and practice guidelines. We maintain a list on the Clinvar website with this URL, so you can check this at any time to find out who's sharing data in Clinvar, how many records they have, and when they last submitted. So each Clinvar record has 4 major domains of data. There's a variant, a condition, and an interpretation of how that variant relates to the condition. The interpretation, as I noted earlier, may also have some related information like a comment that explains the interpretation or maybe citations that were consulted in the interpretation process. The 4th domain is the evidence, and in Clinvar, we structure the evidence as observations of the variant. So the minimal information that we ask for an observation is whether that variant was observed in germline tissue or somatic, whether individuals with the variant were affected with the condition, and then finally the collection method. This is asking whether the data was collected in clinical testing or research or something else. This is a typical structure for data that you'll see in Clinvar. This is how the clinical labs typically submit their data, so this is very common. But you may also see records that have much richer evidence. So for example, we may have a submission from a research laboratory that's studying a family. So they may have a proband as well as mom and dad, and they may be able to provide information about each of those individuals as a distinct observation. And because they have access to the family and they may have consent to share more detailed information, in addition to these minimal fields, they may be able to provide more detailed information like age, sex, zygosity for the variant, any observed clinical features, and their ethnicity and population group. This kind of rich evidence is less common in Clinvar, but it's very valuable when it's available. So once we receive data from our submitters, our main function in Clinvar is to archive that submitted data. So each submitted record is assigned an SCV accession number. And then that accession number also gets a version number so that we can maintain a history of changes to each submitted record. We also aim to standardize the data that's given to us. So wherever we can in submission, we require structured and standardized data. And we also validate that data so that not only the format is correct, but that it's actually meaningful. We also aggregate the data. So as you've seen, we sometimes get data from multiple submitters for the same variant. And as a user, you want to be able to see all of that information together to compare interpretations and compare their evidence. So we aggregate data for the same variant into a record with an accession number beginning with a VCV. We also aggregate at the level of the variant disease pair, and that data gets aggregated into a record with an RCV accession number. Finally, we publish the data weekly, and as a reminder, all of this data is free to access. So it's important to note that the interpretations that you see in Clinvar are community curated, and I'll tell you a little bit more about what I mean by that. So I showed you earlier a variant page where we saw this overall interpretation at the top, and then there's a table further down that shows you each submitted interpretation. So it's probably obvious that the submitted interpretations are really community curations. Each of these labs is a member of the community, and they are submitting to Clinvar the interpretation that they curated for the variant. I think what's less obvious is that the overall interpretation in Clinvar can also be considered community curated. In other words, there's no person at Clinvar who is deciding what this overall interpretation should be. Rather, we use a set of rules to calculate this overall interpretation. And so we calculate it based on the interpretations of that variant that came from our submitters. So for this example, you can see that our submitters told us this variant was either pathogenic or likely pathogenic. And so we have the overall interpretation of pathogenic/likely pathogenic. Our rules also take into account the review status for each submitted interpretation. So you may be familiar with the review status as the stars that you see at the top of a Clinvar record. That's the review status for the aggregate record, but we also give a review status to each submitted record. So you can see in this example, each of these submitted records has a review status of criteria provided, single submitter. So the review status is intended to give you as the user an idea of how much review went into the interpretation and therefore how trustworthy it is. So this is a summary of the review statuses in Clinvar. All the detailed information is available online. The top review statuses are based on who provided the interpretation. So whether the variant was interpreted from a practice guideline or an expert panel. The expert panels are an area where we work in close partnership with ClinGen, the clinical genome resource. They approve all of the expert panels that submit to Clinvar, and in fact, they're developing most of those expert panels. After that, the review status is based on transparency into the rules that a submitter uses to classify variants and evidence for each submitted interpretation. And for aggregate records, we also consider whether there's consensus among submitters for the interpretation. So what this means is that when we calculate this overall interpretation, we are considering the interpretations that came from our submitters, but we're giving a bit more weight to the interpretations that have a higher review status, those that we think probably are more trustworthy. So we make the data available weekly, as I mentioned, and it's accessible in several different ways. You can search for the data interactively on our website. You can download bulk data by FTP. And you can also access the data programmatically using our set of APIs called eUtility. So next, we're going to shift gears a little and talk more specifically about integrating Clinvar data into EHRs to improve patient care. So as with any sort of data integration, integrating genomic data into the EHRs has its own set of challenges. I think the major challenge is the availability of structured genomic data in the EHRs. I believe typically when clinical laboratories provide the report, it's often in the form of a PDF, which is a nicely readable form for humans, but it's not as nicely structured for machines. There can be issues with variable levels of adherence to standards for variant nomenclature in the community. This makes it hard to know that you're talking about the same thing. It's important to know that you have the appropriate level of access to the data that's relevant, and also to make sure that the data is available when it's needed. And then finally, because we're talking about genomic data, of course, there can be privacy issues as well. So for most of these issues, Clinvar can address them using the FAIR principles. So the FAIR principles were developed in 2016 as a set of recommendations for making databases really useful by machines, not just by humans. So the FAIR principles are making data findable, accessible, interoperable, and reusable. And although Clinvar predates the FAIR principles, it actually does follow them. So Clinvar data is findable. Every variant is assigned a unique and persistent identifier. That's the variation ID I mentioned earlier. And the data is indexed so that it's searchable. And we'll talk a little bit more about searching data in Clinvar. Clinvar data are accessible, so our data are available with standard protocols, and the data are freely accessible. Because it's variant level information, not patient level, no authorized access is needed. The data in Clinvar are interoperable, so we use a defined data model that's available for review, And as I mentioned, we use data standards wherever possible, like using HDVS expressions for variants or database identifiers like OMIM, Orphanet, Mondo, or HPOIDs for diseases and phenotypes. And then finally, Clinvar data is reusable. So NCBI does not put limitations on its data usage. And we do maintain a provenance of the data so that that can be tracked as it's used elsewhere. So let's talk a little bit more about how Clinvar data are findable, specifically for use cases that are related to EHRs. So we talked about a couple of use cases earlier, and the second one is really more relevant for those who are using EHRs, the clinicians. So let's say you're a clinician with a patient who has genetic testing results. Some of those questions you might then be looking to ask are how have other labs interpreted that variant, and what phenotypes were observed in those patients. So in that case, you might want to make a very specific query to Clinvar for that exact variant with a query like the gene symbol and the C-dot expression. Other questions you might want to ask are what other variants have been reported at this genomic location? And in that case, you can look by the RS number from dbSNP. So that represents a certain type of variation at a location, so not a specific allele. So you can find out other alleles at that location, which might inform the variant that you're looking at. You may also want to ask what other reported variants result in the same protein change. This can be useful if there's not much information available for your variant. Other variants that result in the same protein change may have a similar effect on the patient. So, for example, you can search for protein changes like V600E. And then finally, you may get results where there's a variant in a gene that doesn't have a particular gene-disease relationship that is known. So you may have to do a broader kind of research to really get into more of a gene-disease question. And in that case, you can ask questions like, what other variants have been reported in this gene? What phenotypes were observed in those patients? So you can search Clinvar for the gene symbol. You will note that all of these kinds of searches really do depend on having that structured genomic data in the EHR, which is why I think that's one of the major challenges for this kind of integration. So thinking about how to actually make that connection between EHRs and Clinvar, one option is for an EHR simply to link out to Clinvar. So again, having that structured data is important if you want to be able to provide your EHR user with information about a specific variant. You can provide a link using something like a Clinvar variation ID or an HDVS expression, an RS number, and take them directly to the specific information that they need. Another use case we talked about earlier is a user may need to have a broader query, tell me all the variants that are in Clinvar for a gene. In that case, it's important to have that structured information for the gene symbol so that you can then query Clinvar for all of the variants in that gene. More information is available on our website as well for other kinds of ways to link out to Clinvar. So in addition to linking out from the EHR into Clinvar, the other option is to incorporate Clinvar data directly into the EHR system. So you remember I said there's two other ways you can access data from Clinvar. You can download data from our FTP site, store it locally, and then present the data that you would like to give to your users in the EHR. Or you can use APIs just to call the data as needed and then present it into the EHR, either way. In each case, I think through this presentation, you've probably thought of some things that you would want to have available in an EHR from Clinvar. A couple of thoughts, or you might want to have that overall interpretation that Clinvar has calculated, and maybe also each submitted interpretation and who submitted it. EHR users might also want to have some information about the quality of the data that they're seeing. So they might want to know how recent is it? When was this variant last interpreted? And they might want to have the review status, that measure of how much review went into the interpretation and maybe how trustworthy it is. And then finally, of course, the clinician looking at an EHR wants to know about what diseases or drug response, what conditions were reported for these interpretations, and maybe also what clinical features were noted in patients with the same variant. So I think there are probably still a lot of opportunities for us to improve this integration between Clinvar and EHRs. One thing that we are thinking about is support for reclassification of variants. So we recently implemented a feature on the Clinvar website for a user to click a button and follow a variant in Clinvar, and then we'll email the user when the interpretation of the variant changes. So right now, that's a manual process, but we could consider developing automated ways to follow a variant so that an EHR system could follow a variant and we could push any changes to the EHR system so the clinician would know when there's new information available. We're also thinking about improvements to Clinvar's data model. One thing that we are considering is providing separate classifications in germline and somatic contexts so that we provide higher quality information in EHRs. Another thing that we are considering are developing APIs that use the FHIR standards. FHIR is the Fast Health Interoperability Resources. These are standards that were created specifically for transferring data between EHRs. Currently, it seems to us that mostly this is focused on transferring patient-level information, and so we're not yet sure if it's really useful for the sort of variant-level information that we have in Clinvar, but it's something that we're keeping an eye on. And then finally, we're very interested in reaching out directly to the EHR vendors to talk about opportunities that we can work together to make this kind of integration easier to do and easier to use for the clinician. Finally, I just want to thank the team who supports Clinvar. On the left is our team of developers, followed by our team of curators, then our leadership team for Clinvar, and then our leadership within the NCBI and the NLM. And finally, I thank all of you for your attention, and I'll be happy to take your questions in the live Q&A.
Bueno, muy bien. Bueno, le agradecemos a Melissa, aunque no pudo estar, pero bueno, estuvo muy interesante. Así que, bueno, vamos a finalizar este bloque y nos encontramos a la 1:30, que tenemos algunas presentaciones más. Así que, bueno, nos vamos todos a almorzar y en 1 hora nos vemos otra vez. Hasta luego.